Hello friends, in continuation to our previous class, today we are going to discuss about C4 cycle, otherwise called dicarboxylic acid cycle or Hatch-Slack pathway. C4 cycle, otherwise called dicarboxylic acid cycle or Hatch-Slack pathway. Before we go into the discussion, we have to learn about photorespiration, because photorespiration and the C4 cycle, even the C3 cycle also are closely related. So understanding photorespiration is essential for further deliberations. What is photorespiration? The name it implied, under the presence of light, there will be a gaseous exchange, just like what we used to do. Say we take oxygen and release carbon dioxide outside. That means under the driving force of light, oxygen will enter and carbon dioxide will release. But for us it is usual, but for plants is a detrimental aspect because it exploits the resources. Photorespiration is not at all a better thing or a desirable thing for the plant. So plants generally try to avoid in one way or the other way through appropriate adaptations. That means because photorespiration leads to no ATP and NADPH or sugar. It doesn't have any biosynthetic capability or biosynthetic phenomenon. Just like C3 cycle ultimately leads, that means it is a biosynthetic phase, because we get glucose, I mean carbohydrates. But here photorespiration is not like that. It's a complete exploitation of resources, because carbon dioxide will be released out at the cost of ATPs. Because carbon dioxide is a raw material for plants, because in photosynthesis it used along with water to synthesize carbohydrates. But here the carbon dioxide releasing out is bearing on the plant. To discuss about this concept, we have to understand the behavior of what we call differential behavior of Rubisco enzyme is must and should. Differential behavior of Rubisco. What we call differential enzymatic behavior. Enzymatic behavior of Rubisco, which is wise to environmental fluctuations. Say high temperature and high oxygen concentration. Rubisco, we have discussed in our previous class, an enzyme that converts, that means used in carbon fixation, say the primary acceptor in C3 cycle, RBB, ribulose 1,5-bisphosphate, to which the carbon dioxide will be condensed to form 2 molecules of 3-phosphoglycerate, which is the first stable product in C3 cycle. Then it further leads to synthesis of carbohydrates. So we have discussed at length in the previous class. But here the case is not the same all the time. I told you, differential enzymatic behavior of Rubisco was advised to environmental fluctuations. Say, when the temperature is high or when the oxygen concentration is high, the Rubisco performs or behave differently. We have discussed in the previous class that there are 2 enzymatic activities. One, carboxylase. 2, oxygenase. Carboxylase and oxygenase are the 2 enzymatic capabilities of Rubisco enzyme. Carboxylase means the diameter implied. It useful in the carboxylation. That means that just like in C3 cycle, the carbon dioxide will be condensed with the RBP to form 2 molecules of 3-phosphoglycerate. Here the conditions are like temperature should be normal, not above. At the same time, there should be an adequate carbon dioxide concentration, because say it's like comparating effect or just like relative concentration of oxygen and carbon dioxide. If carbon dioxide and oxygen more or less are in equal terms, the Rubisco will favor the carbon dioxide. Then the process will form. Rubisco then followed by 3-PGA, then ultimately glucose will be synthesized. But when there is an environmental fluctuation, say concentration of carbon dioxide is low, but there is a high concentration of oxygen or high temperature or sometimes even both, then the case is different. Rubisco resumes its oxygenase activities. Oxygenase activity, the name it implied, it involves oxygen. Say, for example, this is RUBP, then it acts oxygen. The ultimate product is phosphoglycolate, which is a 2-carbon compound. Okay? Whereas in carboxylation phase, RUBP leads to 2 molecules of 3-PGA. That means 3-phosphoglycerate. It ultimately leads to glucose. That means, we have discussed 2 molecules will be used to glucose and some 10 molecules will be less for regeneration. Somehow we'll get glucose, and during this process it's like productive phase, what we call biosynthetic phase. This with respect to carboxylase phase. But oxygenase, I told you already, photorespiration is not at all a desirable process because it exploits the resources. Why? Says carbon dioxide is a raw material, instead of involving in the photosynthesis, it has been releasing. It's like a reversal to the that means the photosynthetic ability, what we call Warburg effect. Warburg effect. Warburg effect simply means reducing the photosynthetic ability by alteration of an enzymatic ability with respect to environmental fluctuations or the parameters. That means here gradually the photosynthetic ability reduced because carbon dioxide has been releasing out instead of utilizing in the production of glucose. That's the Warburg effect. That is how the issue ends. Because of Rubisco, with respect to environment fluctuations, entire phenomenon depends upon. Say for example, the condition is higher oxygen availability, then the RBP combines with oxygen, then phosphoglycerate, and ultimately it releases carbon dioxide by exploiting ATPs. That is why I told you it's about this entire phenomenon is considered as photorespiration. Since the first stable compound, just like phosphoglycerate in case of C3 cycle, here the first compound formed when the oxygen combines with RUBP is phosphoglycerate, and it is 2 carbons, so that it is called C2 cycle. Photorespiration is otherwise called C2 cycle. We will discuss later regarding its further pathway, but the thing here is the differential behavior of RUBP is like triggering effect on the plants to adapt accordingly. That means adapt to the extreme situations. And all this phenomenon is not, it cannot be related to each and every plant. Different plants behave in a different way. For example, we can, for convenience, we can say that the photorespiration generally happens in C3 plants. Say particularly the temperate plants like wheat, barley, etc. C3 plants generally face a problem of photorespiration. Why? Say temperate plants, they cannot adapt to higher temperatures. Higher temperatures, since they cannot adapt, therefore leads to the carbon dioxide release. That means exploitation of resources. But there are certain plants, say C4 plants, which has been adapted to the tropical dry conditions. That means higher temperatures, they are well suited to this condition. That means they safely avoided the exploitation of the resources. How? So before we go into the mechanism of C4 cycle, we have to understand how the C4 plants adapted to this terrific phenomenon. Okay, it's about the basics. So I told you already, C4 plants, it's like there is no other way out. They have to invariably adapt to the extreme adaptations. What are the extreme adaptations? I told you, high temperature. How they adapt? C4 plants adaptations to high temperatures. And high oxygen concentration. so that they can avoid C2 cycle. Into means they can avoid C2 cycle. How? Say there are two major adaptations. Dimorphic chloroplast. Dimorphic chloroplast and Cran's anatomy. Cran's anatomy. This is a granule. This is a granule. Actually, these two are related. Cran's anatomy, C4 plants, for example, sugar cane, the sorghum, the maize. Actually, Hatch-Slack, I have used the word Hatch-Slack. The scientists who have worked out this phenomenon, they try to understand the alternate way of carbon fixation, what we call, carboxylation. And during this process, they have worked on the sugar cane and came to know that there is an alternate mechanism for carbon fixation, what we call beta carboxylation. Hatch-Slack, they have worked out on sugar cane and reported beta carboxylation. It's not that simple. So to understand this process, we have to understand the kind of anatomy. I told you Cran's anatomy. Cran's anatomy, that it means, say this is a normal structure of a leaf. Say in general, I'm trying, okay, for basic understanding. And these are villiform cells. Okay, and the stomata are present here. Just for a rough sketch I am giving to understand how this happens. Then these are vascular bundles. These are vascular bundles. That means that they are nothing but xylem and phloem. Then we knew that mesophyll cells, palisade and spongy. Say these are palisade parenchyma. Yes, I am specifying zone in certain areas because for understanding. And these are spongy. And these are the villiforms, bundle sheet cell. This is very important. Bundle sheet cells. Say these are the mesophyll cells and these are the bundle sheet cells. Then these are vascular bundles. And the thing is, it's about Cran's anatomy. Means in general, there are no specialized cells circulating the vascular bundle. Bundle sheet, the name it implies, surrounding the vascular bundle, just like wreath, just like circular arrangement. Circular element, the red color, there are the bundle sheet cells. And these bundle sheet cells have a greater significant role in C4 plants to avoid the photorespiration. I told you already, the plants tend to avoid the photorespiration because it exploits the resources. And how? I will tell you that. First, they have to adapt accordingly. Adaptation, how? Structural adaptation. Okay, what kind of structural adaptation is the Cran's anatomy? And what is the Cran's anatomy? Development of specialized circular cells around the bundle sheet. That means around the vascular bundle, what we call bundle sheet cells. That means mesophyll cells are normal. Just say, it will say C3 cycle will take place in the mesophyll cell. But in case of bundle sheet cells, the C4 cycle will take place. C4 cycle will take place in bundle sheet cell. What can say? Compartmentalization with respect to extreme adaptations, and this is a typical example to avoid photorespiration. So it's about Kranz anatomy. It means development of specialized cells around the vascular bundles through in the way of bundle sheet cells. Okay. Then dimorphic chloroplast. I told you granule and agranule. Granule means we have discussed about chloroplast in the previous class. So the chloroplast in the internal structure of chloroplast contain thylakoids. Thylakoids are they are nothing but membranous sac flattened structure in the form of pile of coins. They are called as thylakoids, and the arrangement is called grana. Chloroplast with grana are known as granule chloroplast. Chloroplast without this thylakoid arrangement, that means grana, is called as agranule simply. That means only stroma is present. No grama, only stroma with respect to agranule, but just that both stroma plus grana with respect to granule chloroplast. Where they are present? Say, granule chloroplast are present in the mesophyll cells, whereas agranule chloroplast are present in bundle sheath cells. Agranule chloroplast are present in bundle sheath cells. Bundle sheath cells. Plus granule chloroplast present in mesophyll cells. What we call dimorphic chloroplast. Why such kind of element? I told you already. Adaptations to high temperature. What happens? In general, I told you, C3 cycle generally takes place during normal conditions. In normal conditions, carbon dioxide availability is high. For example, say, and this carbon dioxide will be utilized in the carbon fixation, and as usual, the glucose will be synthesized. But during high temperatures, the scenario is different. During high temperature, here what happens? Say these are the mesophyll cells. When the light hits here, when the light falls here, then it enters into the mesophyll cells, and we knew that when there is a grana, then that means there is a chlorophyll pigment. Light reaction will take place. It leads to ATP NADPH. That means non-cyclic photophosphorylation. What we call non-cyclic photophosphorylation. That leads to ATP, NADPH, and all that. Okay, you have discussed already. That leads to oxygen production. This is very important. Oxygen production means gradually it accumulates oxygen. The products of light reaction are ATP, NADPH, and oxygen. When this oxygen concentration is high, I told you, riboscope behave differently. Instead of carbon dioxide, binds with oxygen. So there are two conditions: high temperature and high oxygen conditions behave at like inducing impact on riboscope to behave differently. So to avoid the same, that means to avoid the high temperature, what is the adaptation? Bundle sheath cells. What makes this bundle sheath cells more specialized? Because of its location. Say up to this we can expect higher temperature, because they are the cells facing the light. They are present somewhat inward, that leads to slightly lower in temperature. This zone particularly, and this zone will experience high temperature, whereas somewhat inwards experience some low temperature. At the same time, here in the mesophyll there is a grana, that means the light reaction. Here there only stroma, no grana. So the both conditions having, it's like technical advantage with respect to location, at the same time with respect to structural modifications. The C4 plants has this advantage because they are the bundle sheath cells, which are the site of C4 cycle, are away from the higher temperature because they are inward. At the same time, they have removed the grana, they have only stroma. That means no need of, so there is no dangerous condition with respect to high oxygen concentration or high temperature condition. That is how they avoided this because these two conditions, high temperature and high oxygen concentration, leads to photorespiration, what we call C2 cycle, which exploit the resources. So that is how Kranz anatomy and dimorphic chloroplast evolves what we call a adaptation in C4 plants, which enables them to produce the glucose in a sustainable way, what we call continuity of biosynthetic phase. In case of C4 plants is because of the structural adaptations to higher temperature, say in the way of Kranz anatomy and the dimorphic chloroplast. Very simply, the concept is same. The only thing we have to remember here is just like any other C3 plant, here also in C4 plant the Kelvin cycle will take place, but only point is the location difference. Here the Kelvin cycle takes place in case of bundle sheet cells, whereas the mesophyll cells it takes place in it's like division. Initial carbon fixation will take place in mesophyll cells, whereas the C3 cycle will take place in bundle sheet cells. Why? Because in the C3 cycle the most important enzyme is rubisco, and this rubisco will be active. That means carboxylase activity will be present in bundle sheet cells, whereas in case of mesophyll cells there is no rubisco. Rubisco means here there is other than called PEP case. That means phosphoenol pyruvate carboxylase, PEP case. We'll discuss how it happens. So bundle sheet cells is a significant feature for C4 cell. See, that means C4 plants. That means to enable to successfully run the C3 cycle. That's a connection. The evolution of bundle sheet cells in C4 plants is for continuous synthesis of C3 cycle, and this is present only in C4 plant. That means to distinguish the C3 plant and C4 plant, the first most feature is bundle sheet cells. If we see that if there is a bundle sheet cells, it means that they are C4 plants. And the significance of bundle sheet cells is that the C3 cycle, that means Kelvin cycle, will take place in bundle sheet cells because there is a rubisco enzyme here. That means rubisco with carboxylase activity, whereas in case of mesophyll cells, the initial carbon fixation will take place. But at the same time, the products will be transferred to the bundle sheet cells. I will tell you how it is. Okay? But for now, the bundle sheet cells is the site of C3 cycle. Bundle sheet cells are the site of C3 cycle and is a unique feature of C4 plant. That's a connection. C3 cycle takes place in bundle sheet cells in case of C4 plants, whereas in case of C3 plants, C3 cycle will take place in the mesophyll cells. That's the only difference. The location difference, that's it. Okay, so it's about the base. Then regarding the mechanism, how the C4 cycle will run, we'll discuss about that. Okay, then the name it implies C4 cycle, 4 carbon compound. Just like in C3 cycle, the first stable product of carbon fixation is 3-PGA in C3 cycle. That means 3 carbon 3 phosphoglyceric acid, whereas in case of C4 cycle it is a 4 carbon compound, what we call oxaloacetate. Before we will discuss that means in just like overview. This 4 carbon oxaloacetate is a first stable product, and it converts into some other 4 carbon compounds like malic acid or aspartic acid, depending on the availability of the kind of substrates. For example, if there is availability of amino group, they become aspartic acid, or else they will be converted to malic acid through malic dehydrogenase. And this C4 acid, with all these are formed in the mesophills. I told you already. In case of C4 plants, the Kelvin cycle will take place in bundle sheath, but not entirely. The thing is, the first carbon fixation, that means the initial carbon fixation will take place in the mesophyll cell itself. The product formed, the first stable product, what we call oxaloacetic acid, after it getting converted into some other C4 compound, will transfer into the bundle sheet cells because they are closely arranged now. Through plasmodesmata, the gaps, that means just like the mesophyll cells communicates with the bundle sheet cell through plasmodesmata, and they enter into bundle sheet cells and they continue C3 cycle. Okay, and also eventually regenerate the PEP. Okay, I will tell you how it ends. Say the C4 cycle. Say, I told you already, it will take place in 2 cells, mesophyll cells and bundle sheet cells. These are the mesophyll cells. And these are the bundle sheet cells. Mesophyll cells and bundle sheet cells. And we do that. It's about basic structure, that means cell wall, plasma membrane, and the plasmodespata, you know. This is the plasmodesmata. For understanding, I have signified the same. And this is cell wall. This is plasma membrane. You know. Then it is atmospheric carbon dioxide. Atmospheric carbon dioxide enters into the mesophyll cells as usual. Okay. And this carbon dioxide plus water form carbonic acid H2CO3 by carbonic anhydrase. Carbonic anhydrase. And this again dissociates into HCO3 minus H plus. That means bicarbonate. And this bicarbonate plus PEP, phosphoenol pyruvate. I am writing here PEP, phosphoenol pyruvate. What is PEP here? That means it's like the primary carbon dioxide acceptor. In case of C3 cycle is RUBP. Here the primary carbon dioxide acceptor is PEP, PEP, phosphoenol pyruvate. And this PEP carbon, I told already, what we call initial carbon fixation, what we call primary carbon fixation, and we have discussed. It takes place in mesophyll cells only, not the entire phenomenon. It's like compartmentalization or balkanization with respect to extreme adaptation. Okay, and this PEP. And the enzyme responsible is phosphoenol pyruvate carboxylase, simply pepcase. Say, for understanding, I will write it. Phosphoenol pyruvate. This is structure. And this is after the phosphoenol pyruvate combine leads to oxaloacetate. That means oxaloacetic acid. Take it like this. This is oxaloacetic acid, which is 4 carbon. Okay. This is 3 carbon. You know. Then it is by means of pepcase, it forms oxaloacetic acid. And this oxaloacetic acid become malic acid by malic dehydrogenase. Say here NADPH will be utilized, forming NADP plus. The enzyme here is malic. This I am mentioning as MA, means sorry, MD, malic dehydrogenase. Malic dehydrogenase. For understanding, I will mention the written sketch. Malic dehydrogenase. You know, and after this, sometimes this oxaloacetic acid will also form aspartic acid when there is amine group available, NH2 group available. That's it. And when there is amine group, it becomes aspartic acid or else malic acid. It depends, you know. Then it will transport into the bundle sheet cell. That means the malic acid will transport, which is a 4 carbon compound, transport into the bundle sheet cells. You know what happens next? There is a phenomenon called oxidative decarboxylation. Oxidative decarboxylation. The name it implied. Oxidative means here we remove, say in the way of, you know, we will form NADPH here. because of oxidation. Okay, then decarboxylation, remove carbon dioxide. See here, one NADPH has been formed because of oxidation, and decarboxyl carbon dioxide is formed and ultimately form pyruvic acid. Pyruvic acid, which is a 3-carbon compound. The logic is simple. It's a 4-carbon compound. We're removing carbon dioxide, and at the same time oxidation NADPH is formed. And this carbon dioxide, this is very important. I told you already. The initial fixation will done in mesophyll cells. The next ultimate Kelvin cycle, what we call. That means the whole process of C3 cycle, or the entire process of C3 cycle, will take place in the bundle sheet cells only. But for that, the initial carbon dioxide acceptance, the carbon fixation will be done in mesophyll cell. That's it. So you can say simple, in bottom line is same. C3 cycle in C4 plants takes place in bundle sheet cells, whereas the primary carbon fixation in C4 plants will take place in mesophyll cells. That's it. Okay. And this carbon